Welcome to EPG Pathshala. Today we will be discussing Module 11, Transmission of Genetic Information under the Chapter 2, Structural Organization of Genetic Information in Molecular Genetics Paper 016. Genetic information is carried as long linear biopolymers called as nucleic acids. These represent both DNA and RNA. DNA and RNA are thus defined as long linear biopolymers which are involved in transfer of genetic information from one organism to another. Both DNA and RNA are composed of a sugar, which is a 5-carbon component, ribose in case of RNA, whereas deoxyribose in case of DNA, a base comprising of adenine, thymine, guanine and cytosine in case of DNA, whereas adenine, uracil, guanine and cytosine in case of RNA. Also, there is the presence of phosphate backbone in both RNA and DNA as a third component. So, it is believed that DNA and RNA is made up of a sugar, a phosphate backbone and nitrogenous bases which are universal. The base pairing rules of these biopolymer follows Chargaff's rule. In Chargaff rule, the ratio of purines, that is A adenine and G guanine, is always same as ratio of pyrimidines, that is thymine or uracil in case of RNA and cytosine in case of both. The double helical structure of DNA follows the base pairing, which is a stable hydrogen bonding present in these linear biopolymers. Learning outcomes. We will be going through role of hereditary material in genetic transfer, defining genes, study of differential gene structure of prokaryotes and eukaryotes, studying gene expression, biological information flow, importance of genetic recombination, DNA crossover, meiosis and its phases, importance of meiosis in contributing genetic diversity, understanding classical model of homologous recombination, that is the Holiday model. Carriers of genetic information are both DNA and RNA. DNA and RNA are long biopolymers called nucleic acids, carrying information in the form that can be transmitted from one generation to next. As has been discussed in the introduction, DNA and RNA is the genetic material of all living organisms. The double helical structure of DNA is a resultant of structured base pairing. These base pairs provide a mechanism for copying the genetic information from an existing nucleic acid chain for synthesis of a new chain. A phosphodiester linkage present in macromolecules consists of 3' carbon atom of one sugar molecule linked to 5' carbon of subsequent sugar to a phosphodiester linkage that forms backbone of nucleic acid structure. Base pairing rule by Chargaff. As a base pairing rule, the ratio of purines to pyrimidines is almost equal, that is 1:1. The double helical structure of DNA is result of this structured base pairing. These base pairs also provide a mechanism for copying the genetic information from existing nucleic acid chain for synthesis of a new chain. Gene structure. A gene is defined as a region of DNA carrying information that determines primary structure of proteins through structure of mRNA transcribed. Much of the genetic material of an organism is organized into genes, which are the basic functional units. Genes Specify the kinds of proteins that are made by cells. A prokaryotic gene lacks introns and are often organized in operons that transcribe RNAs encoding multiple proteins. Such genes are said to be polycistronic, whereas a eukaryotic gene is monocistronic and it comprises coding exonic regions interrupted by non-coding introns that are removed by splicing event post-transcription from the pre-mRNA. Gene expression. Gene expression is also called flow of genetic information. It operates with transcription of DNA to mRNA and it is dependent on the genetic code. By definition, a genetic code is arrangement of 3 consecutive nucleotides called as base triplet or codon. This corresponds to 1 amino acid on a protein. Codons in mRNA are read sequentially by transfer RNA molecules. tRNAs serve as adapters in protein synthesis. Protein synthesis takes place on ribosomes, which are complex assemblies of rRNAs and proteins. Prokaryotic gene expression. In a prokaryotic cell, the process of transcription and translation are in close coordination. This is due to lack of nuclear compartment. Eukaryotic gene expression. Since there is a presence of cell nucleus, the site of transcription is usually separated from site of translation, which is cytoplasm in case of eukaryotes. mRNA must be transported out of nucleus into the cytoplasm to find cytoplasmic ribosomes. Initiation complexes and elongation factors bring AAtRNA, which is amino acylated transfer RNA, into the mRNA binded ribosome complex, matching the codon in the mRNA to anticodon in tRNA, thereby adding correct amino acid in the sequence encoding the gene. Upcoming amino acids are linked into a polypeptide chain of resulting proteins. Role of genetic material. It is decoding of genetic information. The genetic material of an organism must carry out 3 essential functions. Primarily, the first function is genotypic function, which is the transmission of genetic information from one generation to other. Second prominent function is the phenotypic function, which directs the growth and development of offspring into reproductive adults. Thirdly is the evolutionary function, which operates through mutations. This allows the organism to evolve in response to changes in the environment. Mutation produces new genetic variability, which provides raw material for evolution. It has also been observed that many specific DNA sequences are conserved among species. This is an affirmation to relatedness of all organisms. Thus, in addition to variation, DNA is also responsible for biological continuity. The mechanism of genetic transmission. Genetic transmission is defined as transfer of genetic information stored in genes to another generation from parent to progeny or from one parent in a cell to other cell. The mechanism of genetic transmission is the working ground for evolution. Genes of most viruses and all higher cells are majorly made up of DNA. As a carrier of genetic information, DNA functions as a blueprint that determines the structure and function of living organisms. Significance of genetic information. Genetic information plays two major roles. First is of hereditary information transfer, which occurs from parent to progeny, or cell-to-cell transfer in asexual organisms, or from individual to individual through germ cells in multicellular organisms. Secondly, genetic information helps maintain cell structure and function in an individual through expressed proteins. Genetic information determines form and quantity of protein produced in a cell. It also determines the localization of protein and its metabolic fate in the cell. Sum total of genetic information of an organism expressed within environmental changes determines phenotype of that organism. The process of meiosis. Meiosis is a form of cell division that results in half the number of chromosome, that is the haploid number, in sex cells or gametes. It is a process of multiplication of germline cells or gametes for sexual propagation of organism. Somatic cells generally divide asexually through a process of mitosis. Genetic information is replicated prior to cell division. Importance of meiosis. Meiosis allows parental chromosomes having same genes called as homologous pair to align side by side and undergo exchange of DNA segment through a process called crossing over. Significance of meiosis is in generating genetic diversity through the process of homologous recombination. Genetic diversity depends upon segregation and independent assortment of allelic combinations. The importance of meiosis is to maintain genome size of sexual organism and to promote variability in genes causing genetic diversity. The process of meiosis. Meiosis as a process of cell division is characterized by two distinguished phases, meiosis I and meiosis II. Meiosis I is a reductional division resulting in two daughter cells with haploid chromosome number, whereas meiosis II is a duplication event of these two haploid daughter cells. It is also called equational division, which finally forms 4 haploid germ cells. Meiosis I. Meiosis I causes reduction of chromosome number from diploid to haploid during the formation of gametes. It comprises of 4 basic phases. Number 1 is called prophase I, which is followed by metaphase I, then anaphase I, and finally telophase I. An interphase stage precedes meiosis I, where the duplication of DNA content of cells occur via the process of DNA replication. Interphase I is a non-dividing stage. During this stage, cell duplicates its nuclear material. Each homologue replicates, forming 2 identical sister chromatids. This results in formation of 2 copies of genetic content termed as chromonema. Prophase I follows interphase 1. It is a complex and elaborate phase of meiosis comprising 5 substages, namely leptotene, zygotene, pachytene, diplotene, diakinesis. Leptotene of prophase 1. It is a short-lived phase representing condensation of chromosomes. Here, sister chromatids of chromosome are distinctly in close approximation seen attached to nuclear envelope. The chromosomes appear beaded due to the presence of interspersed structures called chromomeres. The next stage following leptotene is zygotene. In this stage, pairing of homologous chromosomes called as synapsis starts. Synapsis is mediated by formation of highly ordered zipper-shaped protein structures that are called synaptomial complexes or shortly as SC. Synaptomial complexes have role in converting crossovers into functional chiasmata. Paired homologues form tetrad and they are called as bivalents. Pachytene. Pachytene is a stage after zygotene. Here the synapsis is complete. Recombination nodules are formed at the point of recombination. Crossing over between non-sister chromatids of homologous chromosomes is seen at the point of crossover and it is termed as chiasmata. The two chromatids attached at chiasma exchange parts. This process helps to create and maintain variation in species. Diplotene follows pachytene stage. At this stage, homologous recombination, which is also called as a genetic exchange during crossover, is complete. Synaptomial complex begins to degrade. Chromatids begin to uncoil, revealing terminalization of chiasmata. The last stage of prophase 1 following diplotene is termed as diakinesis. At this stage, chromosomes condense and become thick. Terminalization of chiasmata is complete. Nuclear membrane and nucleolus disappear. Metaphase 1 follows prophase 1. At this stage, tetrads arranged at the equatorial plane. Microtubule strands called spindle fibers from opposite poles of cell attaches the centromere of each homologue. Each chromosome moves independently of all others through a process called independent assortment. This random assortment provides a means of variable chromosome combinations in the future gametes. Anaphase 1 follows metaphase 1. Here, homologous chromosomes separate by contraction of spindle fibers, moving apart towards opposite poles. Sister chromatids of chromosome are intact. Each chromosome at the pole is a monovalent representing a dyad. The last stage of meiosis 1 is termed as telophase 1. At this stage, chromosomes reach the poles. Nuclear membrane appears. Cytokinesis occurs, dividing cytoplasm and organelles between two resulting haploid daughter cells. Meiosis 2. Meiosis 2 separates sister chromatids of each chromosomal dyad. Short interphase of meiosis II lacks synthesis. This event is therefore termed as interkinesis. Prophase II, here the disappearance of nuclear membrane and condensation of chromosome occurs. At metaphase II, spindle fibers attach kinetocores of both chromatids of individual chromosomes at the equatorial plate. At anaphase II, microtubules of spindle fibers contract, thus separating each sister chromatid apart at opposite poles. Each sister chromatid now behaves as independent chromosome. Cytokinesis follows telophase II, forming 4 unique haploid daughter cells, each carrying new combinations of genes on chromosomes. Anomalies in meiosis. Being a detailed process, meiosis is prone to errors. Aneuploidy is the most common chromosomal abnormality caused by non-disjunction of homologues during meiosis I or of sister chromatids during meiosis II. Anomalies in meiotic process can result in chromosomal abnormalities with gain or loss of complete chromosome in the zygote stage. Monosomies, which is the loss of chromosome, and trisomy, which is the gain of chromosome, are lethal except monosomy X, which is also called XO or Turner syndrome. Trisomies such as XXX trisomy, XXY Klinefelter syndrome, and XXY Jacob syndrome result in abnormal and stunted development in the individuals. The molecular basis of recombination through Holiday model. This model was proposed in 1964 by Robin Holliday. This model suggests homologous recombination involving simultaneous single-stranded breaks in 2 double helices of homologous pair which are about to cross over. This model is also called single-stranded break repair model. In general, models of crossing over provide insight about the molecular mechanism of recombination. Starting with the key features of Holiday model. A. The first key feature is enzymatic cleavage. As understood through the figure, in enzymatic cleavage, endonuclease breaks each single strand of homologous pair, that is non-sister chromatids which are about to cross, which maintain same polarity. Both strands are oriented in the same direction, either 3' to 5' or 5' to 3', and these strands are cleaved to create single-stranded break. In step B and C, formation of Holiday junction is seen. In this process, open end of broken strands migrate to join opposite homologous strand. The process is called strand exchange or strand invasion. Ligation of these strands occur, creating a cross bridge called Holiday junction or Holiday structure, with resulting partial heteroduplex. In next two steps, D and E, migration of DNA heteroduplex, also called as branch migration, is seen. Here, Holiday junction displaces downstream, extending the length of heteroduplex. This process of branch migration represents movement of crossover point or migration of cross bridge away from original nick site. It proceeds by breakage and reunion of hydrogen bonds in a zipper fashion. In steps F and G, it is shown that Holiday intermediate can be better viewed by 180-degree rotation of both bottom half structure by the process called as isomerization. Steps H, I, J and K show resolution of Holiday intermediate. This is by the pattern of cleavage which resolves possible crossovers and non-crossover molecules. Through a horizontal cut left to right in heteroduplex after rejoining, the mismatch repair forms a non-crossover recombinant, while a vertical plane, that is up to down splicing or cleavage after reunion, forms crossover recombinants. Actual crossovers are yielded only after mismatch repair of the vertically cut DNA heteroduplex due to reciprocal strand exchange within double-stranded DNA, whereas a horizontal cut after mismatch repair form non-recombinants carrying gene conversions. DNA and RNA are macromolecules involved in transfer of genetic information. RNA is the genetic material for most of the viruses and bacteriophages, whereas DNA is the genetic material of most living organisms on Earth. DNA transfers the genetic material via propagation through parent to progeny or cell-to-cell transfer. The process of transcription involves transfer of information from the DNA template into a functional mRNA, wherein this mRNA transfers the information through translation into proteins. Translation and transcription process go hand in hand for prokaryotes since there is no nuclear membrane present, whereas in case of eukaryotes, both translation and transcription are separated because of presence of nuclear compartment. The transfer of genetic information follows central dogma, which is the basic dogma of biology. It is also called the process of biological information flow. By the process of meiosis, most of the sexual organisms undergo transfer of genetic information, whereas mitosis is the phenomena where basically the cells which are not involved with gametes or non-gametic cells undergo cell division. By definition, meiosis is a process of cell division for gametes or germline cells where haploid cells result. Gametes involved both maternal and paternal components. Meiosis is divided into meiosis I and meiosis II. Meiosis I is also called reductional division, which results in formation of two haploid cells, whereas meiosis II is believed to be equational division with the final product of formations of outcome as 4N or 4 haploid cells. Errors in meiosis can be lethal. Most of the errors in meiosis resulting from monosomy and trisomy results in abnormalities. Holiday model is a classical model to understand the process of mechanism of genetic recombination. It is also called as a single strand break cut model. The process or the pattern of holiday break cut determines the resultant crossovers and non crossovers in recombinants. With this, I end the summary. Thank you.